Hi friends, today is the topic of cell structures, the fundamental unit of life. We will cover the key parts relevant to the human, that is of animal cells, which is some notes on the plants for the comparisons and link into the all nursing applications like cellular damage in the infections or drug actions. By the end of this section, you will be able to describe the structure of cells and apply the knowledge to analyze clinical situations and therapeutic applications. For example, knowing cell membrane functions help in explaining how antibiotics works and why edema occurs in the inflammation. The cell is a fundamental unit of life. Every living thing is made up of one or more cells, and cells are self-replicating structures. The first cells were observed by Robert Hooke and in 1665, and when he looked at the thin slide of code under a microscope and saw a tiny compartment and which is called as cells, like monochromes. Some organisms are unicellular, like bacteria, and while human cells are multicellular with a trillions of specialized cells. In nursing, we have to see about the cellular health is a key. Think about how the chemotherapy targeting the rapid dividing cancer cells and how hypothermia damages cells in the shock. This is of the cell, nucleus and nucleolus and ER, house, Golgi Apparatus and mitochondria is of one of the powerhouse of the cell. Just look into the picture. Human, that is of animal cells, it does not have a cell wall. That's a key difference from the plant and fungal cells. And in plants, the cell wall is made up of cellulose and hemicellulose and proteins. And in fungus, which has the cytoplasm and this is a multi-layer middle lamina for the cell adhesions with the polysaccharides. Primary cell wall, which is flexible, and sometimes the secondary cell wall is stronger with the more cell mass. This rapid walls gives plants the plant structures and reproductions. And in nursing, we focus on animal cells, but understanding this help when studying microbiology, bacterial cell walls, and pycnogenins are targets for the antibiotic like penicillin. Cell wall in the plants and fungi and how its bacteria is a cell wall. The cell membrane, plasma membrane, is a phospholipid bilayer with embedded with the peripheral proteins. This is the famous fluid Moscow model. The model says that phospholipids from the basic structures with hydrolipid heads facing water and hydrophobia tails inside, embedded with the integral protein, act like a channels and which is of transporters or the receptors. For example, insulin and drugs. Peripheral proteins provide support and lymphocyte. This is semi-permeable membrane controls what enters or exits the cell. The nursing membranes damages causes cell swelling in burns and ischemia. Many medications like beta blockers and binding receptor here, the IV fluids affects the osmosis across the membrane. Centrioles or the cylinder structures. Base is made up of 9 triplets microtubules arranged in the cells. At the center is a barrier-like structures for the stability, and there is a cart wheel with a central hub and the 9 spokes connected by the pin heads the microtubules. And centrioles are the pods of centriosome and organize spindle-like fibers during the cell division. There is of mitosis. We can relate it to the nursing how it's of a defect in the centrioles or centrosomes or linked to the cancers, which is our abnormal cell divisions. And many chemotherapy drugs are targeting the mitosis. This is a mitotic, how it's dividing happens, and we can already see the mitotic triplets. These are the pictures. By observing this, we can understand easily. By the pictures, we can understand how it's endoplasmic reticulum, that's ER, how it's performed. The endoplasmic reticulum. This is a network of tubules which is connecting to the nuclear membrane in the eukaryotic cells. Two type of rough ER is present, which has of first is of rough ER, which is a ribosomes attached to the cytoclonic sites and synthesize protein, that is of enzyme hormones. Another one is of smooth ER, which has no ribosomes, synthesize lipids and steroids and detoxifies the ducts and toxics in the liver cells. This is the mind map what we have seen from the first slide to the last slide. Any doubts you can refer to these two books which is of very helpful in learning many aspects of this topic. That's our quick review into the cell structures, and understanding this basic help in pathophysiology and pharmacology and patient care also. Drop your questions in the comment and share with your friends, and don't forget to subscribe to Yaznexaspark for more video. Let's heal and learn together. See you on the next video. Until then, this is Yali signing off. Take care. Bye.